Good afternoon and good morning. There are people from different parts of the world who are attending this training session, and I hope that the first ones were exciting. So people are more and more tuning into these courses that we are trying to arrange online. I'm Dr. Rajas Warke. We have had this theme of sharing knowledge since many years, and this is the new venture that has been going on for a few weeks. Thank you so much, everybody, for being such a stupendous supporter and ensuring that this initiative is actually being taken on a very positive note. Today's subject is going to deal with DNA sequencing, that is next generation sequencing, Sanger, etc. Most of us are from biology background, I'm guessing, or chemistry background. All I want to tell all of you, whoever is listening to this cast, that is the coursework, today's lecture, sequencing is nothing but just few PCRs which are being done. We will try to demystify, as I always call it, demystify how sequencing works and what are the principles behind sequencing working. Since the word sequencing is heard, people think it is something out of the world, but just like space, or I would say aeronautics is actually engineering. So it's aerospace engineering. The same way, sequencing is multiple PCRs in tandem with purification in between. We will try to make this simple enough so that all the students can understand the techniques behind what Dr. Ashish is going to present, and there will be enough assimilation of knowledge. This topic right now is the hottest topic in I would say molecular biology and applied sciences. So everybody is excited to know and wants to know how it actually works, and we will try to make it as simple as possible. Again, I'll try to tell everybody it's simpler than we all think, and if you have any questions, please go ahead. I'll stop here and Dr. Ashish will now start the presentation for today.
Good evening everyone, I am Dr. Ashish. So I am continuing the crash course in molecular biology with taking the topic DNA sequencing, which is going to discuss about the next generation sequencing and majorly in the Sanger sequencing. Okay, so kindly note these dates, following dates, which will be covered in the future for this course. So we are the part of the HiGenoMB, that is HiGX 360, which is giving the service of the sequencing and bioinformatics. So first I will give some introduction about this is basically the DNA sequencing is the process to determine the exact nucleotide of the DNA molecules, and this is after the DNA sequencing invention. It is the basic needs for the foundation of the modern genomics and molecular biology. After the development of 1977 of the Sanger sequencing by the Frederick Sanger and his colleagues, by the dideoxy method in giant termination, it was revolutionized in the field of the agriculture, diagnostic and forensic and personalized medicine. So first automated DNA sequencer was developed in 1986 by the applied biosystem, and then after the Sanger sequencing machine invention in 2001, the draft human genome was completed, which is later on completed by the modern Sanger sequencing in 2003. In 2005, there is next generation sequencing instrument was invented by the Roche team, that is 454, which is based on the pyro sequencing protocol. So now the modern era of the next generation sequencing is started, which covered the short read and long read sequencing. Okay, till the 2020, around 10,000 genome is already submitted in the NCBI database. So as we discussed earlier, there are three generation of the DNA sequencing technology. In the first generation, after the Sanger method invention in 1970 till the capillary jelly electrophoresis invention, it's included in the first generation. In the second generation, the short read sequencing has come, which is Now the Illumina and the ion torrent is taking a bigger role, playing a bigger role in the short read sequencing and the long read sequencing, where we use the nanopore technology by using the direct DNA sequencing without making the shorter fragments of the DNA. So as we discussed in the first generation sequencing, we covered the Sanger sequencing and HiGenoMB sequencing, which is based on basically the chain transmission method and chemical modification method. In the second generation sequencing, the instrument is used for the 454 Solexa, ion torrent, Illumina, okay, and in the PacBio and Oxford nanotechnology based on for the long read sequencing, which is directly we use the DNA for the sequencing. So in the Sanger sequencing, we use either forward or reverse or both primer to generate the up to the 1000 base pair fragments. But in the second generation sequencing, 50 to 500 base pair was prepared with the fragmentation of the whole genome, and then we align to the with the reference genome, and we generate the whole genome sequencing by using the second generation sequencing. But there is gap in the sequences due to the fragmentation of the DNA. So to fulfill the gap, there is third generation has come. It is based on the Oxford nanotechnology, in which we directly use the DNA and pass through the nanopore, and it gives the real time data of the DNA. So as we discussed earlier, as we know everyone, the DNA is made of 4 bases, as adenine, guanine, thymine and cytosine. So determining these molecules in the exact order of the DNA in the DNA sequencing is basically the basic principle of the DNA sequencing. So there are two popular methods in earlier days, that is maximum Gilbert method and chain termination method. So I will play some video. So as we seen in the video, so this is the brief. So as we seen in the video, this is the brief about the maximum Gilbert sequencing. First we phosphorylated the DNA, then we modify the nucleotide by using the different modifying agent. For the A plus Z, we are using DMSO plus formic acid, and for the C we are using hydrazine plus 1.5 NaCl, and for the T plus C we are using again hydrazine, and for the G we are using only DMSO, and for the cleavage purpose we are using piperidine. So after the cleavage, we are getting the different size of fragments of the DNA, and we expose these in the polyacrylamide gel using the X-ray technology. Okay, so as we can see, in the first land there is no A plus Z was found, but in the second land there is C found, and in the T plus C, that is fourth land, we are found C, so it is concluded as C. For the determining the G, we can see in the first land the A plus Z there is slight band, and in the third land there is the G band is clear. So we have determined the G. In the third base pair, as we can see, there is only T was found, where there is no other band was found, so we determine this. So as according to this, we are determining the sequence with the help of the polyacrylamide gel by exposing in the X-ray in the 5' to 3' order sequencing. So thank you for the watching for this video. So now I am giving the basic of the Sanger sequencing, which is majorly used. That is basically the cycle sequencing protocol, which is basically the 7-step protocol. First we denature the DNA, then make multiple copies of the single fragments, then we attach a primer either forward or reverse in one way, and then we add the solution of DD-NTP, that is DDTP and DDATP and DDGTP and DDCTP for each tube. Then we make again the PCR, and then again we denature the DNA. Then we run on the X-ray, and we determine the order of the sequence. This is the older method which have been used for the Sangers. Now in the advanced era, we are adding the fluorescent dye, and we are doing the capillary electrophoresis, in which when the DNA fragments was passed to the capillary, then it gives the signal of the different fragment different signal according to the nucleotide, and then we detect the signals and we determine the order of the nucleotide in the present in the DNA sequence. So the Sanger is a gold standard because the error rate as compared to other sequencing technology, it is very low. There is only one drawback. There is only we can up to 100 to 1500 base pair we can sequence at a one time. So now come to the next generation sequencing technology, which is known as a second generation sequencing technology. It is very cost effective as compared to sequencing. If we are going to sequence the whole genome or exome, and the popular instrument for this is the Illumina and Ion Torrent. The application of the NGS sequencing was frequently used nowadays for the whole genome sequencing of bacteria, fungus and virus, or whole exome sequencing to sequence the protein coding gene. For the whole transcriptome, we are doing the RNA sequencing and determine the differentially expressed gene present in the sample, and we also doing the targeted sequencing to explore the microbial community present in the environmental DNA sample or the clinical sample. We also doing using the NGS technology for the shotgun metagenomics, where we can explore the all microbiome present in the environmental sample or nucleotide sample, including viral, bacterial, fungal, algal, everything we can do by the metagenome sequencing. And for the cancer genomics and rare disease diagnostic, we are focusing the targeted panel and the exome sequencing. So this is the basic protocol to make to prepare the next generation sequencing. And first what we do, we take the DNA and fragmentize it to make the short fragments. Okay, then we make adding the adapters. After adding the adapters, we are doing the PCR, that is called the library PCR, that is the bridge amplification cycles, which is also known as, and we make a cluster of the short reads, and we perform the sequencing using the different type of flow cell available with the Illumina. And after that, we getting the reads, we align the sequence with the reference genome and determine the sequence present in that reference genome. So in case of the genome sequencing, we are covering the all exon and intron part and everything. But in case of the exome sequencing, we are only focusing the protein coding genes in the targeted gene panel. If you are looking for the any targeted regions, that's like BRCA1, BRCA2 or something like that, which is related to any disease or antimicrobial resistance, then we are focusing the targeted gene panel. Okay, but in case of the genome sequencing, the depth is only 30x is recommended to assemble the genome assembly. But if you require the whole genome assembly, including intron exome, then you have to go to higher depth, that is 100x, and you need to also the long read sequencing, which I will later discuss. In the exome sequencing, generally 50 to 100x is recommended to determine the mutation of the protein coding gene. But in case of the targeted gene panel, because the targeted gene panel is mostly popular in the cancer somatic mutation diagnosis, so we need to determine the exact mutation, we need to do the sequencing to more deeper, that is about the up to the 500 to 1000x coverage. So now come to the transcriptome sequencing. It is similar to the whole genome and whole exome, but we start it from the RNA. As we know, the total RNA contains different type of RNA. So major RNA is rRNA, which is not playing the major role for the any disease diagnostic or functional role. So we are targeting the mRNA or circular RNA or small RNA or LNC RNA for the transcriptome sequencing. If you are talking about the whole transcriptome sequencing, then we are talking about the sequencing of mRNA, circular RNA, small RNA or LNC RNA. But some people are also interested to only focus the protein coding genes, that is belongs to the mRNA. So according to that, we are determining the initial protocol. If you are looking for the mRNA, then we are depleting the ribosomal RNA and poly-A tail enrichment was performed to enrich the mRNA. So this is the basic protocol. When we isolated the RNA, then we determine the RIN value by using the Agilent tap station. When we get the RIN up to the 5 to 10, then we are generally recommended for the sequencing, but the type station people recommended the sequencing if the real value is greater than 7 to 10, then taken for the sequencing, for the RNA sequencing. For the small RNA there is no need of fragmentation, as we know the small RNA size is 21 up to the 21 nucleotide to 50 base pair, so there is no need of fragmentation here. But in case of the mRNA sequencing, first we do the poly A tail enrichment, then we fragmentize it, and then we add the adapter. After then we do the cDNA, then construct the library as we perform in the WGS and exome sequencing. After the completion of the, we take the type station value to determine the library size and also take the qubit concentration. So this is third generation sequencing, in which this is basically recommended for the long read sequencing. If you are looking the whole genome sequencing without the fragmentized the DNA, then you have to choose the Oxford nanopore technology, where we put the DNA direct to the nanopore and give the real time data signal of the nucleotide which is present in the ordering of the order of the DNA molecule. Okay, so if you are looking for the whole genome sequencing, then we need to take the hybrid approach, that is short read sequencing and some aspect of long read sequencing. Or if you are looking for some repetitive mutation like Huntington disease, then you have to go for the long read sequencing, because in the short read sequencing there are if the repetitive sequence cannot be detected by the short read sequencing. So this is the major benefit of the third generation sequencing. So this is the application of the NGS, where we can explore the molecular marker biodiscovery for the different type of cancer by using the transcriptome study, and also we can develop the different type of biomarker, antibiotic resistant or something like that by using this study. In the exome sequencing we can understand the rare disease mutation, hereditary genome, hereditary diseases. In the allele mining we can go for the, if you know about the NIPT, in the metagenomics, if you want to explore the presence of the bacterial community in the environmental DNA or clinical samples. Okay, and the epigenetics, if you want to study the methylation of the DNA and their role, then you have to go for the epigenetics. If you want to identify the bacteria, then you have to go for the phylogenetic study by the Sanger or either with the 16S approach. So in the our HiGenoMB, there are all facility we have available with us, that is first generation sequencing, that is Sanger, MISeq and NextSeq 2000, which belongs to second generation sequencing. Then we have also Oxford nanopore, that which belong to the third generation sequencing. So as we know, the NextSeq is 2000 help us to determine the whole genome sequencing and transcriptome sequencing, where we can generate up to the 100 GB data per sample in one run. And the whole exome sequencing, we also choose the NextSeq 2000 for doing the whole exome sequencing. For the microbiome, we either go for the Illumina NextSeq 2000 or the Illumina MISeq, because this is the targeted sequencing based on 16S or RTS. If you are looking for the whole metagenomics, then again you have to go for the either MISeq or NextSeq 2000. Our sequencing law, kindly go through it. So we have the separate sample box, so there is no chances of the contamination, human contamination. This is our area of the library preparation, where we perform the library preparation and our sequencing PCR. So this is our amplicon where we have two separate laminar hood for the preparation of the master mix and separate for adding the DNA template to avoid the contamination. This is the Agilent tap station where we determining the real value or library. We are taking library QC. This is our Sanger machine. This is our server room. This is local server room. We are not uploading the data from the any server. So when you are doing sequencing with us, there your data is secure. Thank you.